It's truly my pleasure to be here today. And before I let you know about the specific research funded by the BCRF, I really want to take a moment to acknowledge and thank again Evelyn for making this possible, for having the vision so that we could be here today. So. Certainly also Larry, and it was great to see some friends, Laura Lasma, Betsy Green today, and to meet some new people. So now to the science. You know, a lot of progress has truly been made in the setting of breast cancer with the development of some targeted approaches that address estrogen receptor HER2, but there's still more work to be done. The way that we're looking at this is that if we can identify the molecular pathways that drive the growth of breast cancer, then we will continue to be able to identify abnormalities in those pathways that then we can target with the development of new agents that eventually will translate into better medical care for each person. The progress today is possible because a lot of people worked before us to get where we are today. And the beauty of what's happening today is that the techniques that have been developed are allowing us the opportunity to take tumor specimens from patients, actually grow them into mice, and then also create long-term living cell lines that mimic what is happening in the patient per se. And by doing that, we can study the mechanisms of growth and also test novel combinations of therapies that eventually will make it into the clinic. You know, the BCRF has been visionary in allowing us the opportunity to have new ideas, put together the teams necessary so that we can do the experiments, not only in the laboratory, but also take these ideas into clinical trials that eventually lead to new treatments. And this is all being as part of certainly the global vision of improving the cure rate for breast cancer, but taking it to the individual patient. So now we have the ability to take these tumors. We can do actually complete gene analysis of the 25,000 genes that are present in the cells. And with the support of the BCRF, we're trying to understand what happens to these genes. Why is it that some genes are active in some patients and not in others? Can we better predict the likelihood that individual patients will benefit from one treatment versus another? So Larry, I'll stop here and we can get into the technical issues, but that's the global approach that we're taking with the support of the BCRF.